Hello, everybody, and welcome to our class on the UCSC Genome Browser. First, we're going to explore the UCSC Genome Browser, and then we're going to load in the same files we loaded into IGV, but there will be some extra steps for UCSC. So we have that in '07, UCSC formatting. And first, what we're going to do is just go to the browser and get familiar with it. And then we're going to try and upload our peak files and bigwigs to look at the raw data like we did in IGV. But using the UCSC browser, one advantage is that there's a ton of data in the UCSC browser that you can overlay in tracks, and we'll sort of go over that. But let's start with the basics of just going to the UCSC Genome Browser. This is what you'll see. One of the first things you might want to do is go into My Data and My Sessions and create an account. I'm logged out here so that this is what it'll look like if you were to try this. And then I have another session where I'm logged in, and we can look at that as well. But you might want to start an account. But really for the basics, you have your genome you can pick out of interest, and you can go to a certain position right away. Or you can just go to the Genome Browser, click on that, and it'll take you right to a place in the human genome that was the default selected. So if we go back there, we could just go right to the human genome by typing in a gene name. For instance, the fire is one we used in IGV, hit go, and now we'll go right to this gene locus. And we'll get into what all this data is, but here you can see the annotation for the gene. And similar to IGV, you can hold down and scroll so until we find the promoter here. So we're close. This is the gene, and the arrows are telling you which direction the gene is going in. And so we can scroll a little further and still further yet. And now we can see the start of this gene. Now let's say we wanted to zoom in a little closer to this first exon. What you can do is touch one of these hash lines here and make a little window by holding down. And then it'll ask you if you want to zoom in. You can zoom in. And now what we can see is this nice several annotations of this gene and some chromatin histone marks to say that it's a promoter, and actually an ENCODE cis-regulatory elements. The red one means it's a promoter here. So this is all good news. We see that this is where the promoter should be for the first exon. And we can load tracks down here that are just by default. You can go through these gene predictions. I have GenCode since we're using that, and NCBI is the gene annotations open right now. Expression, there's a lot of different resources here, but GTEx is a large collection of human tissues at expression levels. So you can quickly look for your favorite gene and see what its expression level is in different things. And this is the ENCODE cis-regulatory elements. Dense means it's very compacted. You can move this all the way to full by clicking on these buttons. You could put in gene enhancers if you wanted, SNPs, human variation, and repeats like we did in IGV. We'll have it here on full. And then if you did change something, all you have to simply do is hit refresh, and now your new tracks will show up. So let's go to that gene expression one of GTEx and see where fire is expressed. So we can just click on this track, and we get a zoom in with a really handy plot of the abundance of this RNA in different tissues. So we can see pretty quick that in cells, EBV transformed in lymphocytes, it's one of its highest expressed cell lines. Turns out this gene actually does work in the immune system and would sort of make sense with this. It's just nice that you can quickly see here some information that this is a non-coding gene RNA, a non-coding RNA, not a protein coding gene. Its coordinates, if you wanted to track it down, its highest median expression, and total median expression, etc. So just some nice summary information already provided for you by UCSC, where it's much harder to import all of this into IGV. And we can look here for these LTRs that we're interested in. We can see there's a MER for ERV1 endogenous retrovirus in the promoter near the promoter region. This is actually probably an enhancer. And these chromatin marks are chip data that we actually downloaded from ENCODE. So we can sort of look at what ENCODE had already produced on the data that we're analyzing. And then one other quick thing here is you can quickly change aspects of each track by right-clicking on this bar here. And so if we wanted to configure this track, we could set its height or its mean. But more importantly, we could say, let's add in, we previously studied K562, let's add in NHEC here, and GM, another T cell line. And then now we will hit submit. And now we can see more colors are here in this histone representing more cell lines and different cell types. And each bar on the side here is going to have different things you can configure, such as colors. And then this dense versus squished, we can see here that we're in full display mode. And if we went to dense, this is going to collapse that information more. So it's just going to say where there's a repeat now and not necessarily all the different family members described. So these are just some things you can configure by right-clicking. If you want to configure this, it's the same way of getting there. You can look in each column here and it'll have different sort of aspects for you depending on what the data is. So the easy thing is just to click on it. You can right click as well. But that is just sort of some general browsing where you enter your gene of interest, go there, you can zoom and scroll, and UCSC will keep you up to date. So that's the main sort of setup, and you can choose all of your favorite information to have here. But one thing I want to show real quick, there's so many tools in here. BLAT is really useful if you want to take the DNA sequence of this site. You can find out where it is in other species and transfer different genomes here. We'll get into that more later in the class. But for right now, in this Genome Browser button, there's a really useful track search here where you can go look for things you're interested in, like RNA-seq or other keywords you can type in here. But if you search for ENCODE, it's kind of nice that they have this whole ENCODE regulation thing here. I'll do that real quick. But you can see that there's actually a track of ENCODE regulation. And yet you could pick each individual track, but there's a lot you can see. There's a ton of data in here. And so I have loaded right now the cis regulatory elements and then the regulation from ENCODE. So we can see what I only picked the K27 acetylation mark, but we could add transcription here and add RNA-seq and maybe TF chip, since we're interested in chip. Now let's see what that track looks like. It'll always end it typically at the bottom of your current browser. And so now we can not only see where the promoter is, but we can go and see that CTCF binds right here. None of these guys have peaks. TRIM22 binds near the promoter fire, CTCF, and it tells you the cell type. So you can see there's a lot of CTCF near the promoter of this gene. So this is really handy. And transcription, we can configure that to only show us, let's say, HepG2, since that's the cell line we're interested in. We can turn all these other guys off. Hit submit. And so now that expression track is just one here. And we can already see that this first exon is made into RNA, and we can see the transcription factors that bind it. And so just very qualitatively and survey-based, you can just kind of look around and see what's going on. So we see that this exon is expressed, but there's not another one in the screen yet, so we'll move it until we find another exon and see if we see RNA-seq associated with it. Okay, here we see some RNA-seq, and there's an exon. You can also zoom out here. So let's say we wanted to zoom out 10x or zoom in forward over a lot or backwards. These are just ways of moving around in here. And so if we sort of zoom out now, you can also move the tracks quite easily. So let's say you wanted to have this right underneath here, you can just slide it up. We can see good expression from these exons. And if we zoom out even more to see the end of the gene, we'll probably start to see more RNA-seq, and zoom out even more. And we can see more RNA-seq throughout here. And then we can look at where it's more expressed. And then now we can see many more transcription factor binding sites since we zoomed out. So that's just a way you can actually use data that's already in here and start to understand something about the promoter region of your gene, or how abundant it is, or how much transcription is coming in the other direction, et cetera, et cetera. So that Genome Browser track search has got a ton of data in it, and you can add whatever you prefer. Now, if we go back over to my data, this is where we're going to start our session of loading our Chip-seq we just ran in here. And so within my data, there's a thing called custom tracks, and that's where we want to start here today. Okay, so you can see it's asking you to upload things. And so let's upload one of our peak files that we had from the IGV lecture. And let's put in an example of this peak. So this is exactly how the file came out of Nextflow. And let's see what happens. So we chose it, and oh no, it can't read it. Huh. Okay, so this is a common issue in data sciences and biology, is that you have to kind of know what format you're in, and file names and types are going to start to be important now. So I did that example because I knew it would fail. And so let's go take a look at what we're looking at here. So UCSC is going to really want a bed file. And here in the class exercise, the basic requirements of a bed file are these three fields. It's the chromosome and the start and stop position. And this is where we'll start to think in ranges, where we're in a range in the genome. You can have a window from coordinate X to coordinate Y, but because there's 23 chromosomes, you have to know the chromosome for that window. Otherwise, that window could be on other chromosomes as well. So it's kind of, and we're going to start to see why that becomes more and more important as we go. But you can make more and more complicated pieces of data inside of a bed file. So here's what our actual max broad peak file looks like. The first line is the chromosome start and end of the peak range, name of the peak, peak score. These are all listed down here below if you don't want to follow around. The strand, a dot means negative, plus means positive strand, so which direction it seems to be going in. But with Chip, you kind of don't really, strand doesn't matter as much because it's a double-stranded event. This is going to give you the fold change at the peak summit, the p-value at the peak summit, and the q value. Statistics we might take a look at later. So UCSC has a couple problems. One, it doesn't know that this is, it knows that it's a bed here, but then it gets freaked out by all these other columns you have here. So you have to kind of tell it what it is. I described a little bit about what we're looking at here. This is a bed 6+3, but really you just have to remember we want to get it into a bed file. And this is the contents and the description that are in that file. But now if we go look, let's take a look at one of our broad peak files. And so you can do that just by dragging it into a text editor, and it'll show up something like this. This is the actual peak file that came out of Nextflow. And here's the problem UCSC is having, is these are contigs in the genome that are not yet aligned to a specific chromosome. It doesn't like that. It wants that chromosome start stop to begin with. And so what we have to do now is delete these. So we have to just delete this. And now we need to give it a header to let it know what type of file this is. And then UCSC can now upload it. So if we take a look at an example here, just again, dragging it into your text editor, So here's a corrected one where we just simply took away all those non-canonical chromosomes, and we added this header here, which is the track type is broad-peak. You'll notice the file ends in .broadpeak. UCSC will know what that is, and then know that this is an interval of a peak. It'll know this is the name of the peak and the score, et cetera, that we just went over. And then you want to name it. So we want to also have UCSC present a name, and it's going to look for it here in the header. And so we're just going to call this ASH2 replicate 2 on the new version of NF Core Chip-seq. So again, I'll just show the same example of running it on old NF Core Chip-seq pipeline versus a new one, and you get the same results as we saw in IGB. So that's really all you have to do. I have that here for you. You can change the names as you want. But once you've deleted those and added this, we can now go back to UCSC and say, choose a file. And let's take ASH2 replicate 1 in the old version and hit submit. And now you can see we have a custom track. That's awesome. So let's go see what it looks like on the browser. Again, it's probably going to want to put it, let's zoom in so this isn't so busy. Again, we'll just zoom in on this promoter region. And so now we can see here, we have this peak file, and we can click on it and see that we're in dense mode. Let's go to full and submit. And really doesn't change much because that's just the peak. So cool. So we see that there's an ASH2 peak right here at the fire promoter, as we saw in IGB as well. And so let's load some more. So we can go to custom tracks. We can just keep adding to this here where we choose a file. So we did ASH1 replicate 1 in the old one, and let's do ASH1 replicate 1 in the new one. Also the old, we have to do this one at a time. Submit. I don't know why this is the case, but sometimes it's best to just hit go and then come back to custom tracks, or it'll forget that you just downloaded one. I'm not sure if it's me or a bug, but it's best just to do that to make sure. Okay, so now let's choose another one here. And so now we'll see that it's keeping track of all of our custom tracks here. So we have ASH2 like 1 old and 1 on the new, replicate 1 on the new, ASH2 replicate 2 on the new one. So we need the old one. So I'm just going to do this for posterity. Come back to custom tracks, add a custom track, choose the file, and now we need replicate 2 old. Here it is. Okay, so we submit that. And now we have old, new for replicate 1 and replicate 2. Okay, cool. Let's go see what that looks like. Oh, good. Okay, so we can see that in the old and the new, we saw a bigger peak of ASH2O in replicate 1 and a smaller peak in replicate 2. But the nice thing is they're identical. So it means that it doesn't matter if you ran on the old or the new, as we saw in the IGV, that, that we can now see that this is very reproducible. What about those bigwigs? They're great to see because they sort of show the raw data. Now let's try, if you try loading one of those, I'm going to skip that. If you try adding a bigwig here, you're going to get an error because it's a very large file. We're sort of, I'll walk you through this here, but we saw that our raw read files, FASTQ files, are 1 to 2 gigs. The BAM is the alignment of saying where that sequence is in the genome that we went over. But it's good to start remembering these names. You would never really want to load this into UCSC, it's huge. But Bigwig is kind of a nice intermediate where it's sort of a summary of the raw data in a lighter format. And so we want that Bigwig. However, UCSC doesn't want that Bigwig in there from your computer because it wants to keep it in cache and in RAM. So what it needs is a URL. And so I provided them for you here. This URL is hosted on Amazon AWS. That's what I prefer to use. This is just an example of the Amazon console where we have a class 2021 folder, and all of these files are in it. And what's really nice about this is I can make this folder public and simply provide UCSC with this URL, and then it can communicate with Amazon and have these tracks. This becomes even more important when we look at last year's class. There's a lot of stuff in here. So you can use that URL structure and just append the name of the file that you're testing in a script. And we'll do that later. But the S3 bucket storage solution on Amazon is super cheap, even for this amount of data. But now we can make track hubs after we've published and have all of these publicly available to other people. Okay, but for now, we're just going to take the ASH2L replicate 1 and replicate 2 bigwigs, and then also the input. It's nice to look at the input as we did in IGV to see, like, are there regions of low coverage, et cetera, and then scaling. So let's go do that. Let's take this guy in this test example, and then you'll do the same thing we just did for these other 3. And I'm going to switch to a more pre-cooked version. So we just put this in. This is a Bigwig file. We hit submit. Boom, it's there. And why was that so fast? It's because UCSC is not actually putting this file into its memory or anything, or storage. It's just saying, oh, I know where that URL is. Now I can go find that data, access it, and then show the data only for the window that the browser is currently on. And that's sort of caching it and makes it much faster. So let's go see what that looks like. Okay, so we got it up here. We have our new Bigwig file. It's in dense mode, and so we're gonna right-click and hit full, and you'll see now we have these sort of raw tracks of our input. Notice the scale here. It's always gonna auto-scale, and this is an important window to know, is that when you click on it, this is what you're going to get is all this stuff. Change it to vertical range, because most, I already know this, but you'll have to just sort of figure it out yourself. The chips are about 19, from 0 to 19, and input is at 25, which is a bit odd. So you're going to want to scale it according to what your chip signal is, so it looks better. And now I've already done this for all 3 of the bigwigs and those URLs I just provided here. And so let's take a look at what we see. Okay, so now what we're seeing here is the fire peak, where we can see the ASH1 replicate 1 had a bigger peak, both in the old and the new, and the replicate 2 had a smaller peak, but all in the same spot. And Max provided us with a consensus sequence is in a bed file already, and you can just upload it in that BWA merge library Max folder from the output of Nextflow. And so here's our raw data. We can see that kind of makes sense that there'd be a peak here, and we can sort of zoom out and see what's going on around the edges, and that looks like a nice clear peak that's consistent across all the replicates. And if we look here at the input, there's almost nothing. So in IGV, we didn't really rescale anything, but we can see here that this is set to a max of about 1.4, whereas, so that means that there's only 1 read, whereas this track here is set to 19. So if we had set the input to 19, where the chip signal is, to keep it on the same scale, you can basically see that there's no signal. And so now our input track looks empty. And so that's cool. It means there's some nice peaks, and those other ones did not quite make it here because there wasn't enough signal over random. We now have this lined up with all of that information. It's repeat maskers. There's a promoter, right, where there's supposed to be a promoter. This all makes sense. You can look at the expression, add in that RNA-seq track if we wanted to. But those were the main concepts. That UCSC is great because you're gonna have so much data at your hands, but you're gonna have to think a little bit about what UCSC wants and its formatting. And you can load peak files from your computer because they're very light and not very large files. But the bigwigs, you're going to want to have somewhere on a local server where there's public access to some folder. And I'm sure your IT group has some form of this already set up. Okay, so I think you are ready to explore UCSC. Check out some of these other mirrors. Or one last thing I want to say is that what's great about UCSC is its output figures are wonderful. So you can say view PDF PostScript. I always like PostScript. So you can download the image if you want of the chromosome, but we want the graphic in a PDF. And so now we'll have a PDF of this, which we can put into Illustrator. It's in vector graphics, and we can make it look really nice and pretty really quick. And so with really running this through Nextflow and of course Chip-seq pipeline, you got all the output files you needed. For lab meeting, you can probably present all the QC results, some quick browser shots, and things like this. So it's really just a couple things you have to think about, and you can get really far into already starting to make figures. And we can suggest here this gene we like a lot, that ASH2L is at its promoter very, very specifically, or very well represented at its promoter. Okay, so I think that with that, we will leave it to more exploration. You can Google what some of these other tracks are, put them up, and have a look. There's some really cool disease tracks in there, but really it's endless in UCSC with the resources you can have. Just a little bit trickier to get your data in there, but once it's in there and organized, you're good to go. And you can go into My Data now that you're logged in and say My Sessions, and it's going to have a default of HG38. And I'll just say for today, I'll call this class video overview. And now when I save this session, I just hit submit. And now it's there. I have this session, I can go to it. I've had other things I've been wanting to do. So you can have this whole collection of different views, different genome versions. There's different data and different genome versions. So you might want to save sessions for that. So I recommend getting an account and logging in right away and saving your sessions. And once in a while going through and cleaning them up. It's pretty easy to delete them. So with that, I hope you enjoy the UCSC Genome Browser and looking at your Chip-seq data in there and seeing what you see. Until then, code light.